তবে এটা তো তোমরা জানো যে তোমাদের নেক্সট সেমিস্টার অনলাইনে হচ্ছে, এটা শিওর আর কি। এক্সেপ্ট ল্যাব কোর্স, মানে যদি হচ্ছে ভার্সিটি খোলে তারপরেও কিন্তু কোর্স অনলাইনেই হবে। কোর্সে সব মানে ফিজিক্যালি হবে না, শুধু হচ্ছে তোমার কি বলে প্র্যাকটিক্যাল কোর্সটা নেওয়ার প্ল্যান আছে আর কি আমাদের, বাকি সব কোর্স অনলাইনে। ঢাকার বাইরে যারা আছো তারা প্র্যাকটিক্যাল কোর্স নেওয়ার আগে কিন্তু চিন্তা ভাবনা করে নিও যে একটা কোর্সের জন্য ঢাকায় আসবা কি আসবে না।
স্যার ওই যে আমার তো স্প্রিং এ ল্যাব 4 ছিল, ওটা তো
হুম হুম
অর্ধেকই
হুম
আটকে গেছে ফাইনালের জন্য, এখন কি আমাকে
হুম
যাইতে
হুম।
হবে?
আচ্ছা, ওইটার ফাইনাল না নিয়ে আমরা রেজাল্ট দিতে পারবো না আর কি।
এখন কি
ওকে।
স্যার আমি ওই একটা কোর্সের জন্য ঢাকা যাব?
না, যেটা হচ্ছে ওই কোর্সগুলো যেগুলো করা হবে, এটা তো স্প্রিং কোর্স, স্প্রিং সেমিস্টারের কথা বলতেছো না?
হ্যাঁ স্যার, স্প্রিং 20।
আচ্ছা, এগুলার ক্ষেত্রে যেটা করা হবে, ধরো যেই কয়টা এক্সপেরিমেন্ট বাকি আছে
হ্যাঁ
এগুলো একটু মানে ওই এরকম না যে প্রতি সপ্তাহে একটা করে, অথবা টানা ওগুলো করায় দিয়ে এক্সাম নিয়ে জাস্ট গ্রেড দিয়ে দেওয়া হবে, এমন না যে ওইটার জন্য তোমার অনেকদিন ঢাকা থাকতে হবে।
আচ্ছা আচ্ছা। তাহলে যেটা স্যার, ফ্যাকাল্টি তো চেঞ্জ হয়েছে ল্যাব 4 এ আর 3 তে,
হুম হুম
তাহলে
হুম।
কি এটা কি যে নতুন ফ্যাকাল্টি যাদের অ্যাসাইন করা হয়েছে ওরাই
উম,
নেবে ফাইনাল?
না, আগের ফ্যাকাল্টি মানে যারা অরিজিনালি অ্যাসাইন ছিল আর কি। কারণ আমরা তো এখন ওই 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 স্প্রিং সেমিস্টারের প্র্যাকটিক্যাল কোনো কোর্সের রেজাল্টই দিইনি।
হ্যাঁ হ্যাঁ হ্যাঁ
সো ধরো আমি তো একটা নিতাম সেকশন, সো ওইটা তো আমার রেসপন্সিবিলিটি, নতুন ফ্যাকাল্টির তো না।
হ্যাঁ হ্যাঁ হ্যাঁ।
সো ওই আগের আগের গুলো আগের ফ্যাকাল্টি নিবে আর কি। এবং এমন না যে ওইটা হচ্ছে তোমার যে প্রতি সপ্তাহে যে একটা করে ক্লাস হতো ওইভাবে হবে না। ওটা ধরো
আচ্ছা,
দ্রুত
একটা
মানে
ডেজিগনেটেড টাইম দিয়ে দিবে, ওর মধ্যে ল্যাব হয়ে যাবে।
হ্যাঁ শেষ করে ফাইনাল দিয়ে আমরা গ্রেডটা দিয়ে দিব আর কি।
আচ্ছা, এটা হলে স্যার মনে হয় ভালো হয়।
ওটাই করা হবে, কারণ তোমার নতুন কোর্স তো শুরু হবে, পুরাতন এবং নতুন আমরা তো অ্যাকোমোডেটই করতে পারবো না, এত তো ল্যাব নেই আমাদের।
সেটাই।
আচ্ছা। আচ্ছা, আমরা একটু আগের লেকচার গুলো থেকে কোনো কোয়েশ্চেন আছে? সহজ লেকচার, I mean, if you have any question from the previous lectures and until lecture 12, not 12, lecture 14.
Sir, is there any change in our final exam date?
No, I don't think so. So the final exam was scheduled for 29. Right?
Yes, sir.
Yeah. So we didn't change the date, and I think we will not change the date.
Okay, sir.
So because changing the date will be extremely difficult.
Okay, sir.
So if anyone have any problem means like say you have another exam in the same time, please let me know. Okay. The exam will be on 29. It will not be changed, but if anyone have that kind of problem, so then at least let me know. Okay. Okay. So if you don't have any question from previous lectures, I meant until lecture 14, then today we are gonna discuss about lecture 15 and 16, and then we are done with the syllabus. The good part is, I mean, about lecture 15 and 16, most of these things you are already very familiar. So it will not take long time, and also it will be very easy for you to read these lectures. Okay. So first, lecture 15. Lecture 15 is about the microbiological tests. So what kind of I mean, what we need to test and what kind of tests are done regarding the quality check in food and beverage industry. And not only food and beverage industry, basically the strategies or types of tests are similar in pharmaceutical industry and food industry in context of microbiological quality. Okay. So if we think about the other hazards like the physical or chemical, tests will be different. Okay, so it will differ between the food and pharmaceutical. Okay, some tests will be similar, but some tests will be different. But if we think about the microbiological tests, those are kind of similar. Okay, so I mean, what is the purpose of doing microbiological test? We all know, right? Because if spoilers or pathogenic microorganisms are in the food, then they will cause trouble. I mean, if spoilers organisms are present, they will spoil the food. If pathogenic organisms are present, they can cause diseases to the consumers. Okay. So, and okay, there are lots of organisms out there, right? So, do we detect every organisms when we check the quality of food? No, it's not possible, right? So, there are some selected microorganisms. So, these selected microorganisms vary between food to food depending on the raw material. Okay. So, however, these two slides they name the usual microbes which are usually tested in food. Okay, to confirm the quality, such as coliform, E. coli, Salmonella, Bacillus, Listeria, okay, Pseudomonas, yeast, mold. Okay, so these are the these are not all. Okay, just keep it in mind. These are not the all. Okay, these are some examples bacteria which are most frequently tested in food. So depending on the raw material, the test, I mean, there are some more microbes are added in the list. Say, for example, if we harvest the raw materials from, like, say, potato or honey, okay, so this kind of crops may contain a spore of Clostridium species, okay, or a spore from other spore forming bacteria, especially many soil bacteria. They produce spores. So if we harvest our raw materials directly from the soil, okay, in that case, it is necessary to also check for the spores, different mycotoxins, okay, and how we detect or check these microbes, the old traditional methods, which we are all familiar with, like split plate technique, pore plate technique, membrane filtration techniques, MPN method, most probable number method. Okay, so there are also some rapid tests out there, but to check, confirm the presence of microorganisms, culture is still a gold standard. Okay, so different rapid tests are sometimes done, but microbiological tests, I mean, culturing is must. Okay. Okay, now let's talk about the tests which are done in the food industry and also in the pharmaceutical industry. So, what are the basic tests? Pyrogen test, then, like, okay, pyrogen test, sterility test, then preservative efficacy test, then container integrity test, growth promotion test. Okay, so these are the basic testings which are done in a food and pharmaceutical industry. Okay, so first, pyrogen test. So what is pyrogen test? We all know. Pyrogen tests are done to detect the endotoxin. Okay, and lal test is still now the gold method. I mean, we can say this is the gold standard method to detect endotoxin, okay, in the product. Okay, and why it is named lal test? Because the reagent uh, I mean, the reagent that is used to check the pyrogen, its name is Limulus amebocyte lysate, LAL. Okay, so from this name, the test named as lal test, and this chemical is harvested from horseshoe crabs. Okay, this crab, natural source. So the blood of this crab is blue colored, and their blood contain this chemical. Okay, and this chemical can form clot when they interact with the endotoxin. Okay, so these reagents are very widely used in the testing laboratories, in the pharmaceutical and food industry. food laboratories. Okay, so especially in the pharmaceutical laboratories where endotoxin testing is must. Okay, so now I think you all know about the pyrogen test, right? So we
Yes,
have
sir. We pharmaceutical পড়ানো হয়েছিল।
Yes.
আচ্ছা ফার্মাসিউটিক্যালে ছিল তো, সো সবাই তোমরা তো জানোই এগুলা। সো pyrogen test, and you also know about this horseshoe crab. However, you know that because of heavy harvesting, horseshoe crab is becoming an endangered animal. Okay, it's not animal. Okay, it's becoming endangered. So that's why people are looking for alternative. Okay, so some alternative have already been invented by different research group. However, till now, this natural source is most famous. Okay, because although there are reagents, not harvested from the horseshoe crab, but rather synthesized in the laboratory, but still this somehow these reagents are not getting that popularity. Okay, but well, those group, they are trying to make this synthesized reagent to get famous. Okay, so if they become successful in doing so, in that case we will be no longer need the horseshoe crab. Okay, so basically this information I just collected from the Wikipedia. Okay, so in Wikipedia, if you search horseshoe crab, you will get the page, and there you will get this information. Okay, so basically this it contains the information about those synthetic reagents by which we can do the lal testing. Okay, so basically what they did, I mean, in this chemical, okay, so the lal chemical, the whole chemical is not necessary to form the clot with blood, with the endotoxin. Rather, this part, RFC, limulus clotting factor C, only this part of the lal reagent is required to form clot with the endotoxin. So if we can synthesize this factor, we can directly use as a reagent. Okay, exactly that they did, this group. Okay, this group from the National University of Singapore. Okay, they are able to synthesize the RFC factor. Okay, and also another group from the Lonza. Lonza is a big pharmaceutical company. Okay, Lonza is a big research group. Okay, so they also developed synthetic RFC by which can be, which is produced by Eli Lilly and Company. Eli Lilly is a very big pharmaceutical company. Okay. Anyway, so that was about the pyrogen test and how we do the pyrogen testing. Very simple. Take the liquid that you want to check for endotoxin, add the lal reagent, and then incubate at 37 degrees Celsius for 1 hour. Okay. Then if you just invert the tube like shown here, if clot is formed, you will find the clot in the bottom of the tube. Okay, so if clot is there, then it will confirm the presence of endotoxin in that test liquid. Okay, so the advantage of this testing method is simple, easy to perform, takes only short time, hardly 1 to 2 hours to finish the total experiments. We don't use the animal. Okay, not directly, indirectly we use, right? We are making the horseshoe crab endangered. Oh, sorry. The limitations, tests should be conducted with lots of precaution in order to avoid the contamination of the sample. Okay, so now another type of testings which is done in the for quality control purpose. Okay, sterility testing. So anyone want to ask anything?
স্যার
Yeah.
হ্যালো
হ্যাঁ হ্যাঁ শুনতেছি।
হ্যাঁ স্যার ফার্মাসিউটিক্যালসেও তো আমরা লাল টেস্ট পাইরোজেন টেস্ট এগুলো পড়ছিলাম তো ওই
হুম, একই
হ্যাঁ
জিনিস।
ওই
Same.
ওই মানে ওই রকম ইন্ডাস্ট্রির সাথে ফুড ইন্ডাস্ট্রি বা এরকম কোয়ালিটি মানে ওটা তো ছিল মেডিসিন ইন্ডাস্ট্রি, তো বেসিক কোনো ডিফারেন্স
হুম।
আছে?
আচ্ছা, টেস্টে কোনো বেসিক ডিফারেন্স নাই। হ্যাঁ, কারণ হচ্ছে ওই পাইরোজেন টেস্ট করার জন্য একটাই মেথড মানে ইউজ করা হয় আর কি। ওকে, সো টেস্ট মানে এক্সপেরিমেন্টাল প্রসিডিউরে কোনো ডিফারেন্স নাই। তাহলে ডিফারেন্স হচ্ছে কোন প্রোডাক্টগুলো চেক করা হয়। এখন নর্মালি যে ফুড আইটেমে সব ফুডের কিন্তু পাইরোজেন টেস্ট করার দরকার হয় না, স্পেশালি বেবি ফুডের দরকার হয়। কারণ বেবি ফুডে পাইরোজেন মানে পাইরোজেন কি, পাইরোজেন তো আসলে এন্ডোটক্সিন আর কি। বেবি ফুডে এন্ডোটক্সিন থাকলে সেটা তো একটা প্রবলেম আর কি। ওকে, বাট টেস্টিং শুনে যে
ওকে স্যার।
মাইক্রোবায়োলজিক্যাল যে টেস্টিং মেথড এগুলা কিন্তু সব জায়গাতেই সেম। ধরো তুমি এখন হচ্ছে ওয়াটার কোয়ালিটি টেস্ট করবে, ফার্মাসিউটিক্যালের জন্য করো বা ফুড ইন্ডাস্ট্রিজের জন্য করো, সেম প্রসিডিউর হবে। একটা ডিফারেন্স হতে পারে ফিনিশ প্রোডাক্ট টেস্টের সময়। কারণ ধরো ফিনিশ প্রোডাক্ট টেস্ট করার আগে কিছু প্রিপারেশনের দরকার আছে। সো ফর এক্সাম্পল তুমি ট্যাবলেট থেকে মাইক্রোঅর্গানিজম ডিটেক্ট করতে চাও, ট্যাবলেটটা তো আমাদেরকে গুঁড়া করতে হবে, রাইট? উই হ্যাভ টু মেক ইট পাউডার, দেন উই ক্যান ডু দ্য টেস্টিং। এখন ফুড আইটেমের উপর ডিপেন্ড করে, মানে প্রোডাক্টের উপর ডিপেন্ড করে ওই ইনিশিয়াল প্রিপারেশনটা একটু ডিফারেন্ট হতে পারে, বাট তারপরে মেথড অ্যান্ড ম্যাটেরিয়াল সেম। ওকে, okay, so now the sterility testing. Okay, we need to perform this test to ensure that the items were steriled as desired after the sterilization process was performed. Okay, right. So not all the things or ingredients need to be sterile in the industry, right? So especially sterility testing is very important for the pharmaceutical industry, because in pharmaceutical many pharmaceutical preparations they need to be sterile. Okay, so some food items also need to be sterile. Well, let me clear you one thing, like when we say the word that sterile means the I mean usually we understand that sterile means it will be completely free from all kind of organisms, but in industry this word is used differently. Industrial sterility. Okay, if you search this word you will find that the meaning of industrial sterility is different than our usual understanding. Okay, because in the industry, say for example for a food item, say for example milk. Okay, so we don't want that the milk contain any microbes. We don't want that, because if spoilers microbes are present in the milk, they will spoil the milk, right? If pathogenic microbes are present in the milk, they will spoil, they may cause disease to the consumer. But in industry, it's almost impossible to kill, I mean, make them completely sterile. Okay, because it's a food item. If we do very harsh treatment, it will it may interfere with the quality of the food. It will deteriorate the quality of the food. খাবারের quality নষ্ট করবে. At the same time, to achieve that level of sterility, the cost will be very high. Okay, so that's why in industry, rather than the usual sterility, they use industrial sterility term. Okay, so basically industrial sterility means, say for example, if you want to define it for food item, so the food item can contain a certain amount of bacteria if it is safe for the consumer, and also will not degrade the quality of the food if stored as mentioned. Okay, মানে ইন্ডাস্ট্রিয়াল স্টেরিলিটির ক্ষেত্রে হয় কি যে ফুড আইটেম থেকে মাইক্রো অর্গানিজম কমপ্লিটলি রিমুভ করে এটা অলমোস্ট ইম্পসিবল আর কি ইন্ডাস্ট্রিতে। সো সেখানে ইন্ডাস্ট্রিতেও ইন্ডাস্ট্রির ইন্টেনশনটা থাকে স্টেরিলাইজেশন প্রসিডিউর, যে মাইক্রোবসের সংখ্যা এমন পরিমাণ কমায় আনা হবে যাতে সেটা হচ্ছে হিউম্যানের জন্য সেফ থাকে, কনজিউমারের জন্য সেফ থাকে, আর হচ্ছে যে ওই মানে যে ওই অ্যামাউন্ট অফ মাইক্রো অর্গানিজমটা ওই ফুডটাকে স্পয়েল করবে না, তাকে যদি আমরা তার ডেজিগনেটেড স্টোরেজ কন্ডিশনে স্টোরিং করি। ওকে, ওকে, সো বেসিক্যালি by sterility testing we can check that. We can check whether, I mean, it's a for pharmaceutical products. Some pharmaceutical products need to be completely sterile, say ophthalmic products. Okay, or say for example, you are checking the sterility of the food item. So it's not necessary that the food item need to be completely sterile, but if it follow the industrial sterility parameter, then it will be fine. So whatever our goal is, we can check those by sterility testing. Okay. And usually sterility testing, the methods, there are two types of methods: membrane filtration method, direct inoculation method. Okay. Objectives, easy to understand. Anyway. Okay. So for sterility testing, depending on our goal, we will use different types of media. Say for example, if we want to detect aerobes, we can use peptone broth or glucose peptone broth. For anaerobes, cooked meat medium, semi-fluid meat medium. Okay. For detecting both aerobes and anaerobes, okay, we can use this kind of medium, thioglycolate broth media, etc. Okay. And for detection of aerobic and lower fungi, soybean casein digest media. Okay. So okay, there are many different sorts of media, and depending on our target microbes, we can choose our culture media. Okay. And about the test method, we already heard that either we can test by membrane filtration method or by direct inoculation method. So what we do in case of membrane filtration method, first we filter out our test samples, then we incubate the filter on the media. Okay. Say for example, you want to test water, or say you have produced a juice, and that juice is consumed by the baby, so it have to be sterile, say. Okay. So if we want to test liquid products, then using membrane filtration is a better choice, because the thing is, I mean, usually in the liquid products, the amount of bacteria is low. Okay. So if we use direct inoculation method, there is a high chance that we may lose, I mean, we will not be able to detect the bacteria. Okay. So membrane filtration is a better choice for liquid food products. So what we do, first we use a filter paper. So filter papers have different pore size. So which filter paper will we use? Again, it depends on the purpose, that which microbes we want to, which size microbes we want to detect. Okay. But anyway, so in this method, we'll first filter out the liquid product, and during this process, the organisms, they will get stuck on the filter paper. Then we'll incubate that filter paper on media. Okay. Then after incubation, we'll observe the colony, and through further analysis, we can detect what microbes they contain. Okay. Well, but theoretically, we can also use a specific media here, right? So if we know that these microorganisms we want to detect. Okay. So in the industry, usually they use the indicator microbes. So they already know which microbes they want to detect. So it's better to use selective media for the industry, right? Because we already know which microbes we are going to detect. Okay. Okay. So depending on the characteristics of microbes, an additional step can be added here. Say for example, there are some fastidious microbes. Okay, growth of fastidious microbes directly on media is sometimes, I mean, sometimes they don't want to grow. So sometimes enrichment may are necessary. So if enrichment necessary, what we have to do after filtration, first we have to, I mean, use this filter. I mean, first we have to allow this filter to grow in some enriched media. Okay, then we can incubate onto the plate. Okay. So it depends on the microbes, what we want to detect. So usually the test methods are same. Oh, I mean, in the laboratory for, I mean, whatever the laboratory setup, in the research laboratory or in the industry, the basic test methods are same. Okay, now direct inoculation. So for direct inoculation, we'll directly inoculate our sample onto the media. Okay. So in case of industry, what they can do. Say, for example, you want to check your liquid raw material, okay, and the liquid raw material is in the test container. So for first, you have to collect a certain amount of that liquid from the container and mix with a liquid media. Okay, then mix them properly. Then you have to incubate a sample from that liquid, and plate them onto the plate. Okay, simple thing. However, we need to incubate it for 14 days. It is important. Okay, so usually in the laboratory, most of the time we incubate for 1 or 2 days, but here we have to incubate them for 14 days because food items may contain many slow growing bacteria. Okay, many slow growing bacteria which can spoil the food. Okay, so to ensure that we don't miss to detect the slow growing bacteria, the incubation period is longer, for 14 days, sometimes even for 21 days. Okay. Okay. Okay, interpretation of result. So how we interpret growth of organisms, you all know, right? Okay. Now the thing is, especially for the raw materials used in the industry, there is an approach that, okay, so say you do the sterility test, and after 14 days you find out that the raw materials contain undesirable bacteria. So will we discard that raw material? No. We have to repeat the experiment again. Okay. So if in the repeat test we find that we find that bacteria, then we'll discard that. So if in the repeat test we don't find that same bacteria, then actually we have to again repeat that test. Okay. And based on the result of the third test, we'll decide whether we are going to discard the raw materials or not. Okay. Okay, so advantage of mm, okay, just 1 second. Okay, so advantage of direct inoculation is wide application for mostly all kind of products. A very large volume can be tested. A smaller volume of growth required. However, there are some disadvantages. Okay, we need a skilled worker. Cost is high, and another problem is disadvantage of this method is volume constraint. Okay, so volume constraint actually a problem for any large volume raw materials, because say the raw materials are in usually larger containers. So there is a chance that there are bacteria, but they are not on that place from where we are collecting the sample. This is called volume constraint. মানে এই যে কন্টেনারগুলোতে থাকে, কন্টেনারগুলো তো ছোট না, এগুলো বড়। সো লিকুইড প্রোডাক্ট তো এরকম হওয়ার কিন্তু সম্ভাবনা থাকে যে হয়তোবা আমরা যেখান থেকে স্যাম্পল নিচ্ছি সেখানে ব্যাকটেরিয়া মাইক্রো অর্গানিজম নেই, অন্য জায়গায় আছে। ওকে, সো এইটা হচ্ছে এই মেথডের একটা ডিসঅ্যাডভান্টেজ আর কি। ওকে, যে আমরা আসলে স্যাম্পলিং কোথা থেকে করতেছি, এটা একটা ইম্পর্টেন্ট ফ্যাক্টর, যে আমরা স্যাম্পল কালেকশন করার সময় কোন জায়গা থেকে করতেছি আর কি। ওকে, so anyway, and okay, okay, I missed that line. And also inherently bacteriostatic or fungistatic compounds cannot be removed or adequately neutralized. Okay, so the raw materials, they may contain some static material, so like say bacteriostatic materials, so they will inhibit the growth of that bacteria. Okay, so if the raw materials contain this kind of static materials, we may get a false negative results. So false negative results means like we will fail to detect the bacteria, but once the raw material are used for processing the food, they may start to grow again. Okay, I mean, in the raw material container, because of the presence of the bacteriostatic or fungistatic components, their growth was prevented. Okay, but once when we'll use this raw material for food preparation, the concentration of these chemicals will go down there. Okay, so then the microbes will be able to again grow, which may spoil or cause problem in the final product. Okay. Okay, now then another type of test done in the food industry. Preservative efficacy test. So the food items as well as pharmaceutical items, they contain many different types of preservatives. Okay, so we need to test whether our preservative is effective enough to prevent the growth of the microbes at least until the expiration date. Okay, and expiration date পর্যন্ত যদি গ্রোথ প্রিভেন্ট করতে পারে থিওরিটিক্যালি সেটাই এনাফ। হ্যাঁ, কারণ এক্সপায়ারেশন ডেটের পরে গ্রো করলে সেটা তো ইন্ডাস্ট্রি কিছু করার নেই, তারা তো বলেই দিছে এক্সপায়ারেশন ডেট হচ্ছে এটা। ওকে, so at least the preservative have to be efficient enough to prevent the growth until the expiration date. Okay, well, this slide names some of the preservatives used in the industry. You can just have a look. Now the test method, I mean how preservative efficiency test is done. Okay, okay, so do this test. What we have to do, we have to prepare the samples by ourselves. Okay, we have to prepare the test sample, and every sample we and those test sample we have to mix microbes. Okay, so basically the method is done like this way. We have to prepare total 5 say test tube. Okay, and in each of these test tube we have to add defined microbes. Okay, so usually for the test in 3 tubes we add 3 different indicator bacteria, and in 2 tubes we add indicator fungi, and the concentration of the microbes in the tube should be like from between 10 to the power 5 to 10 to the power 6. Okay, then we have to add the preservative there, and then have to incubate them for 28 days. Okay, and we have to check them on different days, like at day 7, day 40, day 21, and day 28. Okay, 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 so this photo just show, I mean, how we'll check that sample on these days, 7, 14, 21, 28 days. Okay, so this is the test method to check the efficacy of preservative. Okay, so now another type of test means that these are the samples, basically the environmental monitor. The environment of the plant need to be continuously monitored. So whether the environment continually, and here the environment means the air, the surface of the walls, floors, different like the furnitures. Okay, so this is environmental monitoring, and environmental monitorings are done by these test methods: contact plate method, swabbing method, settle plate method. Okay, so contact plate method means we'll say, for example, we want to collect sample from the floor, okay, or from the wall. So we'll just take a plate which contains solid agar plate, and we'll directly contact or touch this plate with that surface. Okay, this is contact plate method. We'll directly touch the plate with the surface from where we want to detect the presence of microbes. Okay, so this is contact plate method. Okay, the advantage of contact plate method is we can use any type of media. Okay, and it's actually a very easy method. Okay, but the disadvantage is we cannot collect from everywhere using this method. Okay, and sometimes I mean, we cannot collect samples from every time, from every kind of places, and also in case of contact place, okay, only a tiny amount of microbes they get transferred from that, like say from the wall to the plate, okay, only a tiny amount, okay. So there is a high chance we may miss those microbes which are present in a very small amount, okay. And also colony overgrowth may happen. Okay, so wall contain, I mean, those places contain different types of microbes. Some are very slow growing, some are very fast growing, some may present in very high amount, some may present in very low amount. Okay, so especially the fast growing and high amount, I mean, which are present there in high amount, these microbes cause the problem. Okay, they most of the time they cause overgrowth, so it's become difficult to detect. Okay. Okay, so it was contact plate method, then swabbing method. Okay, in swabbing method, we'll use swab, like say for example cotton bud. Okay, so I mean in this case we'll use swab to take sample from the place. Okay, and then it is same. Okay, just the sampling method is different in this method. I mean contact plate and swabbing. Only the difference is in the sampling method. Okay. I mean, in this case, we'll directly touch the media with the surface, but in swabbing method, we'll use a swab to collect the media. Okay, then finally, settle plate method. So what is settle plate method? So for example, you want to check the air quality, okay, or you want to detect the presence of microorganisms in the air. So just take a plate, okay, with a solid media, or you can also use the liquid media here. Okay, just take your media, put it somewhere. Say for example you want to check air quality in your room. Okay, so put it somewhere in your room and just keep the cover open for a certain period of time. I mean you can just keep the cover open for 1 hour maybe. Okay, then after that incubate your sample for 24 hours and look for growth in the next day. Okay. So advantage by this method, we can check the air contamination, easy to conduct, cost effective. Okay, and only viable organisms are detected. The disadvantages is still air in undisturbed rooms. Okay, I mean if there is no air circulation and the air is still, this method will not be a very good one. Okay. Still air don't tend to get settled that quickly. Okay, then container integrity testing. Okay, so container integrity testings are required to check the containers which we'll use to say fill our product. Okay, and also okay. So like say for example, you are producing juice, so you will use containers to package your juice, right? So then you have to check the quality of that bottle, right? So now if you use raw materials for another industry, yet you have to use some maybe bigger container. Okay. So whatever the type of container, they need to be checked. Okay. So how the container integrity testing is done. Okay. Okay, so what we have to do first, initially validated by filling container with sterile growth medium, then inserting container in broth containing 10^6 CFU per ml of suitable microorganism. Okay, so containers sealed under a vacuum should be periodically tested to demonstrate that vacuum is maintained over shelf life. procedures in place to detect faulty containers during manufacture. Operators involved in visual inspection should have frequent breaks, regular eyesight test. Okay, so basically what we have to do is say we have to check a container. We have to add sterile media there. Okay, we have to just add sterile media in the container. Then we have to collect the sterile media and let them grow through incubation. Okay, so if we observe growth of microbes, then it means these microbes have get contaminated from the container. That means the containers are contaminated. Okay. Now, finally, growth promotion testing. Okay, so growth promotion testing is necessary to check the quality of the reagents that we use. Okay, I mean, in the testing mentioned here, like the sterility testing or the air quality testing, whatever, we use different kind of media, various medias. In the industry, I mean, well, we definitely purchase the media from uh, defined companies, from certified companies. But still, we have to check the quality of those media before we use them for detecting microbes in our plant. Okay, so this is called growth promotion testing. So we have to check whether, I mean, if our media is really capable to support the growth of that organisms which we detect. Okay, আমরা যে মাইক্রোবস গুলো ডিটেক্ট করা হয় কোয়ালিটি কন্ট্রোল ল্যাবরেটরিতে, এই মিডিয়া যে মিডিয়াটা আমরা ইউজ করতেছি সেটা আসলেই ক্যাপাবল ওই ডিটেকশনের জন্য এই গ্রোথ প্রমোশন টেস্ট দিয়ে ওই কাজটাই করা হয়। ওকে, okay, so how we can check this? Okay, it's a simple method. Okay, so we just have to make plate, I mean broth or agar using the say we want to check the media. Okay, so we have to make agar media or broth media using that media, and then we have to add a defined amount of microbes with that media. Okay, then we just have to incubate them and observe whether growth occurs or not. Okay. Okay, that's all for this lecture 15. Anyone have any question from lecture 15? আচ্ছা, আমরা যে মিডিয়াগুলো ইউজ মানে আমরা যে টেস্টগুলো করতেছি, টেস্ট কিন্তু সব জায়গায় সেম। হ্যাঁ, টেস্টের কোনো ডিফারেন্স নেই। আমরা ল্যাবরেটরিতে যে টেস্ট করি, তারপর হচ্ছে ইন্ডাস্ট্রিতে ডায়াগনস্টিক সেন্টার হতে পারে, রিসার্চ ইনস্টিটিউশন, টেস্ট মেথড সবসময় এক। টেস্ট মেথডে কখনো আসলে কোনো ডিফারেন্স হয় না। ওকে, জাস্ট অ্যাপ্লিকেশনটা ডিফারেন্ট হয়, যে আমরা কি পারপোজে ইউজ করতেছি। কোন ইন্ডাস্ট্রিতে হচ্ছে কোন স্যাম্পলগুলো আমাদের টেস্ট করতে হবে, কোন কোন জায়গার স্যাম্পল টেস্ট করতে হবে, এইটাই ইম্পর্টেন্ট। টেস্ট মেথডটা আসলে সবই সেম। আর এখানে যে টেস্ট, আই মিন দ্য টেস্ট মেথডস ডেসক্রাইবড হিয়ার, দিস আর অনলি ফর দ্য microbiology lab, not for the chemistry lab. The chemistry lab, they have to perform other experiment to like check the presence of I mean like exotoxins, and also presence of heavy metals, okay, melamines, etc., mycotoxins. Toxin detection, I mean usually toxin detection is done by chemistry lab, and they do it especially by using SPLC, chromatographic, high profile liquid chromatography, and basically they use the chromatographic method. In your undergrad, and you'll not learn SPLC, but don't worry. In higher study, I mean, you may. It depends. Okay, so if your lab work with SPLC, then you will have the chance to learn it. If not, then you will not get the chance. যাই হোক, হতাশ হওয়ার কিছু নাই। হ্যাঁ, দুনিয়াতে হাজার হাজার টেস্ট আছে, মানে অসংখ্য টেস্ট আছে। সব টেস্ট কি আর একজনের পক্ষে শেখা সম্ভব নাকি? Actually, lecture 16টা একটু ওভারঅল একটু আইডিয়া দিয়েই দিই। হ্যাঁ, lecture 16 is খুব ছোট। I mean, lecture 16 is a small lecture. Okay, so if I can give a brief about lecture 16, then we will be done with the syllabus today. Okay. So lecture 16 is about quality control of beverages. So what is beverages? We all know. Okay. So just read the history by yourself. Carbonated beverage was invented in 1766. Wow, right? Okay, so anyway, I mean, if we try to categorize the beverages, basically beverages fall into two categories: alcoholic beverage and alcoholic beverage. However, soft drinks like say cokes, juice, people usually don't refer them as non-alcoholic beverages. People just tell them soft drinks. Okay, call them soft drinks. Okay, so usually non-alcoholic beverage means, well, you can find non-alcoholic wine in the market if you look for. Okay, so usually those kind of beverages from where which were subjected to de-alcoholized are called non-alcoholic beverages. মার্কেটে এরকম পাওয়া যায়, হ্যাঁ, যে ওয়াইন বিয়ার পাওয়া যায় যেগুলোর মধ্যে কোনো অ্যালকোহল থাকে না। বেসিক্যালি এগুলা থেকে মানে ডিউরিং দ্য প্রোডাকশন প্রসেস, those wines or beers usually contain the alcohol, but they are removed. They are a removal process to remove alcohol from the liquid. Okay. এরকম drinks পাওয়া যায় আর কি। So basically those kind of drinks are referred as non-alcoholic beverages. Okay, so anyway, I mean, if we think about the quality control of beverages, we have to think about both the quality of raw materials and finished product quality. Okay, so we have to check the quality of raw materials as well as the finished product. Okay, so first, then it is important to know what kind of ingredients are used in beverage production: water, carbon dioxide, sugar, artificial sweetener, preservative, color, acids, flavors, okay, etc. So these kind of ingredients are used to make ingredients. So to make the beverages. So depending on the raw materials, there are different test procedures. Okay, because say for example for water, there are at least 62 parameters which need to be tested before we can use the water as an ingredient. Okay, so I mean, if they want to use, I mean, beverages, water is one of the main part of the beverages. Okay, and if we want to use a water to formulate our beverage, there are 62 parameters which need to be checked. Okay, and these parameters actually include microbiological, physical, chemical, every kind of things. Okay, and WHO has a guideline. Okay, regarding the drinking water quality, and US FDA, they have guideline for the water which can be used for beverage industry. Well, actually, US FDA have guidelines for every kind of ingredients. Okay. Okay. So from this information, you can, like say for water, 62 parameters need to be checked. So different ingredients, there are different parameters which we need to check. Okay. So then carbon dioxide. Okay, so major source of carbon dioxide are natural or synthetic byproduct of petroleum refinery. Okay, also byproduct of carbon dioxide are produced as a byproduct of brewing industry. Brewing মানে হচ্ছে alcohol production industry আর কি। Okay, and the commercial available carbon dioxides, they are found in three forms, either in solid, dry ice, liquid. Okay, or gas. Okay. Dry ice is quite frequently used. I mean, like say, not for transportation. I mean, sometimes dry ice are used for transportation purposes. Okay. অলসো আচ্ছা ওই যে বিভিন্ন কনসার্টে দেখা যায় না ধোঁয়া উঠতে থাকে ওগুলো কিন্তু ড্রাই আইস দিয়ে করা হয় ওকে sometimes you may see that in different cultural programs okay smokes are there so this kind of smokes are made by using dry ice okay anyway then sugar artificial sweeteners okay so the sugar that the food industry will use they should have certain criteria okay such as the chemical standards of sugars or moisture should be less than 0.1% and an amount of residual sulfur should be less than 15 milligram per kg okay and there are also uh, microbial limit number of yeast should be less than 10 mold less than 10 mesophilic bacteria 200 per 10 gram of sample okay and in case of industry sugar does not mean the sugar we used in our home okay so in our home we use the sugar made from sugar cane but in industry the majority sugar source is generated from corn actually okay corn syrup ভুট্টা থেকে বানানো হয় কি হ্যাঁ ইন্ডাস্ট্রিতে যে ভুট্টা থেকে যেই কার্বোহাইড্রেটটা পাওয়া যায় এটা একটা মেজর সোর্স ইন্ডাস্ট্রিয়াল সুগারের ওকে সো আমরা বাসায় যে সুগারগুলো ইউজ করি সেটার ক্রাইটেরিয়া আর এই ইন্ডাস্ট্রিয়াল সুগারের ক্রাইটেরিয়া ডেফিনেটলি আলাদা হবে কারণ সোর্সটাই কিন্তু আলাদা এবং প্রিপারেশনের প্রসেসও আলাদা the industrial sugar I mean which is made from corn they may contain many yeast mold and bacteria okay so that's why there is accepted limit that if the amount of yeast or mold is less than this then the food industry can use that otherwise no okay artificial sweetener okay usually they are prohibited for using but they have some limited permission I mean food which are produced for diabetic patients they can use artificial sweetener but not for regular uses okay okay preservative color acid flavors so there are actually criteria for every kind of things especially for food industry and pharmaceutical industry these criteria are strictly maintained okay I'm not sure about Bangladesh but they should be strictly maintained okay if we just go through this these are very easy thing to understand okay and other than this raw materials there are other equipments or materials which are also used for soft drinks say for example packaging materials so crown caps cans pet bottles these can be used so also the quality of these things need to be checked before they are used in the industry okay okay so now which are the common microflora usually the soft drinks contain okay so these are the example of common microflora which may be present in the soft drinks okay so that's why a special focus is needed to detect for this common common microorganisms okay so let's see what are those common microflora typical spoilage microorganisms okay and usually these typical spoilage microorganisms they cause macroscopically detectable changes okay I mean the changes done by a spoilage organism they're easily visible so say they will change the uh, color of the product they will produce a very off flavor okay they may produce change the texture of the product okay I mean the changes will be macroscopic so what are those spoilage organisms they can be yeast or bacteria or mold like yeast, a strong fermenter, Saccharomyces, Candida, film yeast, PCA, weak fermenter. Okay, and from the bacteria, Lactobacillus, Leuconostoc. Okay, and there are also other organisms which spoil the food, but they don't cause macroscopic changes. Okay, so the changes are microscopic. So there are some mold, Aspergillus, Penicillium, they can cause microscopic changes. I mean, to detect those changes, we need to do some testing. We just cannot, I mean, easily sense those changes. Okay, acetic acid bacteria, ubiquitous flora, some okay, some other bacteria related with public health. Okay. Okay, so these are the common flora of soft drinks. Well, soft drinks may also contain contaminated microbes. Okay, contaminated from uh, okay, so contaminated from the drains or the waste product. So that's why the soft drinks they also should have a sanitary index. So what sanitary index means? I mean, what sanitization is done in the industry? Okay, so like say to remove microbes from the floor, okay, so from the even the toilets. So there is a chance that the food microbes can get contaminated by say fecal organisms. So if sanitization is not properly done in the industry, food may contain fecal contamination. Okay, so the food should also, there is a sanitary index that the food should not contain any coliform or E. coli, Enterobacter, Streptococcus, etc. Or if the food contain, there are expected limit that the okay. Usually, all the food items they cannot contain any coliform bacteria. Sorry, any fecal coliform. A minimum level of coliform can be present, but no fecal coliform. Okay. And then finally, food poisoning index. So this index is used to confirm that whether this food contain any spore or toxin. So for example, Clostridium. Okay, this kind of bacteria they produce a spore. Okay. So if the food contain a spore of this bacteria, they can then germinate in later period and become vegetative and produce toxin. Okay. Okay, so these are the common flora. Then what kind of test is done to check the beverages? Okay, so the test procedure are same for soft drinks, non-alcoholic beverages, also water. Okay, so we need to test the this parameters like the sugar content, colors, vitamins, ions, organic acids, ethanol, caffeine, gluconic acid, etc. Presence of, we have to test presence of residual chemicals such as heavy metals, pesticides, mycotoxins. Okay, we have to test the package. We have to test for foodborne parasites, and in addition to this, there are various miscellaneous tests like oxygen sensitivity of beverages, assessment of flavors. Okay, microbiological stability. Basically, this is related with the preservation. Okay, species-specific identification of beverage spoiling microorganisms, etc. Okay, that's all for lecture 16. Anyone have any question? এই কারো কোনো কোয়েশ্চেন থাকলে করতে পারো আর কি। We are done. Syllabus শেষ। So we are done with Mike 403. I mean, the syllabus is finished. We have the remaining final exam and also assignment. আচ্ছা,
স্যার আমাদের ফাইনালি কি মিডের সিলেবাস থাকবে?
না মিডের সিলেবাস থাকবে না, নো মিড সিলেবাস। Okay, if you guys don't have any question, then I think we can stop the class. আমি ধরে নিচ্ছি কারো কোনো কোয়েশ্চেন নাই, তাহলে আজকের ক্লাসটা শেষ করে দিই। ওকে
স্যার
ওকে
স্যার, হ্যাঁ বলেন।
স্যার মিডের খাতা কবে দিবেন?
মিডের খাতা এই উইকেন্ডের মধ্যে পেয়ে যাবো আর কি, মানে কিছু
আচ্ছা
খাতা
স্যার।
দেখা বাকি আছে তো। আচ্ছা, যেটা আমি করব তোমাদেরকে আমি হচ্ছে ওই, হ্যাঁ উইকেন্ডের মধ্যেই পেয়ে যাবা, খাতাও পেয়ে যাবা, এই মার্কও পেয়ে যাবা আর কি। এই খাতাগুলো ইন্ডিভিজুয়ালি দেওয়া একটু টাইম কনজিউমিং, আর ইন্ডিভিজুয়ালি শেয়ার করতে হয় আর কি। যাই হোক, তবে উইকেন্ডের মধ্যে পেয়ে যাবো অবশ্যই। সো নেক্সট ক্লাসে ওই মিডের কোয়েশ্চেন নিয়ে যাহা দাবি দেওয়া আছে সবই আলোচনা করতে পারবা, ডোন্ট ওয়ারি। ওকে, ঠিক আছে, আল্লাহ হাফেজ, আসসালামু আলাইকুম। হ্যাঁ, কি সাকিব কিছু বলবো নাকি?
না স্যার, ঠিক আছে, ওকে।
হ্যাঁ, ওকে ঠিক আছে।